for conditions like ALS and schizophrenia, and we're going to discuss how GLP-1 medications could affect the brain. Lots of exciting, buzzy topics. We really appreciate you being here with us tonight. One very important disclaimer for those of you who follow the TV show White Lotus, the season finale is tonight. So this is your reminder, do not go on social media, and if you see something, do not tell your friends in the room. It's very important that we don't spread any of that around. The other important housekeeping note is that we do take audience questions, and we really want to hear from you. You will see me or someone else from STAT walking around with a microphone during the sessions, so please flag me down. We take these questions throughout the interview, and there's no need to wait until the end, so don't be shy because we really want to hear from you. Lastly, I want to say a very big thank you to our presenting sponsor, AbbVie. With that, we're ready to dive in for our first session of the evening, A Scientist versus ALS. Please welcome to the stage Dr. Merit Sakovich, Executive Director of the Mass General Brigham Neuroscience Institute, Director of the Sean M. Healy and AMG Center for ALS at Mass General Hospital, and the Julianne Dorn Professor of Neurology at Harvard Medical School, and to moderate, STAT Senior Writer for Biotech, Adam Feuerstein.
Hey, everyone. So let's just take a moment to acknowledge the incredible San Diego weather.
Yeah, I don't know why we live in Boston.
Yeah, I'll admit, I spent about 2 hours at the pool today. Not that you could tell, because I'm still pasty white Boston guy, but yeah, it's nice weather here. Yeah, and as we're all sitting here, like I said, some people are watching the White Lotus tonight. As people know, I cover the intersection of biotech and Wall Street, so a lot of my readers are freaking out about the markets, imploding tomorrow. So,
yeah,
but we're relaxed and we're gonna. Yeah. So I'm gonna call you Merit.
Yes, please.
Because we've known each other for a long time, and so you're here. You actually got a Lifetime Achievement Award today. So congratulations.
Thank you. Thank you.
Now, I don't know about you, but when I hear someone gets a Lifetime Achievement Award, that's sort of like.
Yeah.
You know what I mean? Like, you're not going anywhere, right?
No, I want it to be a midlife kind of award. But
perfect.
Yeah.
But it does bring up a question because ALS, obviously, I think everyone knows how devastating a disease ALS is, and also how frustrating it has been for drug developers, for physicians who treat ALS patients just by the lack of, effective medicines. And I wonder, why do you do this? What motivates you for so many years now doing this? What motivates you to continue to try to seek new treatments for ALS?
For sure, the patients are the motivation. I mean, it's in neurology one of the worst illnesses, and the patients are so amazing. They're young, and they really want to work together to cure it. And even though we haven't had that many successes, we have some. And as long as we keep learning from it and not do the same thing, I think we'll finally crack this illness, hopefully in our lifetimes.
So tell us a little bit about your speech today and your talk today, and what the themes were that you were speaking to the group about.
So the themes were that right now for ALS, there's a whole world community that's working together to solve it. And if you looked at it 10 years ago, there were maybe just a handful of people working on it. I think the Ice Bucket Challenge and just technology and science has changed that. And so there's a global effort to conquer this. And the patient voice has largely been missing in it until recently. And it makes it just more exciting and I think faster and better. So the theme was that, together we can kind of conquer this. And the second theme was that just the revolution in genetics and the ability to turn on or off genes in the brain is having a big impact in ALS and neurology. Mm-hmm. And the best example is our one drug for a familial form of it, where we've actually seen people, not everybody, but some people stop progressing and get better.
Right, so that's Qalsody,
yes.
The drug for Biogen. Yeah. So what is it about, when you think about that drug, because it is one of the success stories of recent years in ALS, and granted, for what percentage of patients are amenable to treatment with Qalsody?
So it's small. It's 2%.
2%.
Yeah.
But it's still, just finding a drug that targets a sort of that genetic, specific genetic cause. Talk to me a little bit about that and like what that meant and like where is that sort of a springboard, do you think, for future drug development?
Yeah, I think we learned a lot from that. It's a springboard because it's given us a potential biomarker. So in that trial, we saw this blood and spinal fluid marker neurofilament go down, months before we saw the clinical effect. And we know from other fields like MS that as soon as you have a marker, whether it's imaging or blood, that predicts clinical success, the field explodes. So that might be our first one. It's not perfect, but it's one. And then it also showed us that if you treat early, you can have a big effect. And while we thought about that hypothetically, we actually didn't have proof of it. So those two things are just, it kind of woke up the field. Like finally said, well, actually we can stop this illness.
And how many genes are tied to ALS right now? Like what do we know about sort of the genetic underpinnings of ALS?
So if we look at everybody with ALS, about 20% of people have a single gene that might be causing it. And there's at least 20 of those. And there's probably 40 to 50 genes that are more like risk modifiers. They just increase your risk, but they're not guaranteed to give it to you.
So as the development of Qalsody, has that spurred more investment in sort of looking at other genetic causes or being able to find treatments that would be targeting those genetic causes?
Yes. So there's now a phase 3 trial with Ionis of an antisense oligo or gene therapy for a different form called FUS. And so that actually is a global study because it's pretty rare. It can be juvenile onset, so people in their teens, and it's very aggressive. And so there's hope for that. And then there's a whole effort at Columbia University by Neil Schneider called Silence ALS, where for the even rarer ones, they're kind of partnering with a nonprofit and LORB to make gene therapies for those families too.
I don't know if there's a simple way to answer this question, but why has ALS been so difficult to crack as a disease?
Yeah, it is a difficult illness to crack, and a couple of reasons. One, I think, is we're getting to patients too late. So from the first symptom that someone has, which is probably, after they've had the illness for some time, it takes a year to get to a diagnosis. So, and then they only have a lifespan, maybe 2 or 3 years. So we're getting to them very late. So that's one problem. And then the other is, for all these neurodegenerative diseases, having the tools to know what's going on in the brain, in the spinal cord. Sometimes we look at cancer, how they're getting all their successes, but they have the ability to get the tissue and see what's going on. We don't have that in brain diseases. And that, that's a big, it slows down drug development.
So the diagnosis thing it's an interesting question because that seems like a healthcare structural issue, right? I mean, doctors are not recognizing the symptomology of ALS early enough. How do you fix that issue to get earlier diagnosis?
I think there's 2 ways, or maybe there's more than 2, but one is structural, is about education and using maybe AI or other tools to flag it for primary care doctors. I compare it like if I saw a person with a rare cardiac disease, I probably would miss it too. If a primary care doctor has never seen someone with ALS, they might not think about that. So getting to the doctors where the patients are first going to and giving them tools to think about ALS. And so there is this initiative that the ALS Association started to help with that. It's called Think ALS, but it's pretty much telling primary care doctors what are the first signs so that they don't send someone to the orthopedic or the ENT, send them to the neurologist.
And ALS, I guess, is a disease such that if you aren't diagnosing it fast enough, patients are they just become too advanced?
I think that's a big barrier for the trials.
For clinical trials.
Yeah, that we're starting late in their illness. And we know that again from that gene therapy, the Qalsody trial, that people did better who started earlier compared to those later. Yeah.
As you sort of look out at the landscape of ALS, because obviously you see everything. I imagine there isn't a drug or what I don't know if you want to categorize it in particular areas, but like maybe give us a little bit of a snapshot of what the pipeline looks like today for ALS.
So the pipeline's big. There's over 300 companies in the space. Not everybody's ready for human testing, but there's a lot of great ideas out there. First of all, I would say, having run about 30 studies and not having many that have been successful, I'm not sure I'm the right one to make the prediction, but I keep trying. One of the fundamental biology problems in ALS is this particular protein, TDP-43, where it should be in the nucleus and it's not of the cell, so it's in the wrong place and it's aggregated. And there's a fair amount of data that if you can get it back where it should be in the nucleus to do the right job, that might be really transformational. And 98% of people with ALS have a problem with that protein. So there's several companies that are targeting that biology that hopefully will come to trial in 2025.
So you mentioned neurofilament as a biomarker, which seems to be a really promising biomarker in ALS. When you think about drug development, and particularly early stage drug development, where you're trying to find like a signal, a sort of proof of concept. I mean, is it the biomarkers, or are they the key for those sort of clinical trials?
I think so. What's happened in ALS and other areas in neurology is because it's difficult to have those biomarkers, people kind of skip that early development phase of drug development, and they go right to the pivotal trial, which means that we're going in not 100% sure on the dose and hitting the target. So I think that now that we have a few biomarkers, neurofilament being the best one, spending more time in that early phase and making sure your drug is doing what it's supposed to do might increase our odds of success in phase 3.
Is that a question of the severity of the disease and sort of the paucity of treatments that companies want to sort of move quickly, kind of just jump from phase 1 to phase 3? I mean, are you sort of saying tap the brakes a little bit?
Yes. Yeah, I mean, being not the one who's raising the money or doing it, it's easy for me to say, but that seems to be one of our major issues. And part of it's driven by that there is a pathway by the FDA for single study approval for serious illnesses. So, so sometimes companies want to go for broke, right? They want to go for that. And it's hard to get investment for that earlier phase to do the actual work of the biomarker development. But I think one lesson we have in ALS is we really have to go back to that kind of basics of clinical trial and do that early development work.
I should say, I know Catherine mentioned, if there's any questions, if anyone has a question, raise your hand and we can, but I'll continue if you guys don't. So I'll talk to you a little bit about the Healy platform, because that's kind of what you're known for at Mass General, and it's had some mixed success. As you said, I mean, a lot of these drugs failed. I mean, what have you learned through that platform trial, and particularly how, moving forward, changes that you're making to that sort of maybe to sort of come up with better signals of activity in drugs?
Yeah, I wanted to first share that when we were designing this platform trial, the statistician asked me, what did I think was the prior probability of any drug being successful? Asked a bunch of us. And given the state of the knowledge of ALS at that time, we all said 10%. So we've done 7. So I think I have to do 3 more to get that success. No, but I think that's why I think we're going to have better success if we go in with a higher probability of success by doing that early studies. But nevertheless, we started this platform trial. We've tested 7 drugs, of which none of them hit the primary outcome measure. So that's important. But some of them had some effects on the secondaries that are now going forward to phase 3 testing. What we learned is we need to go longer in our trials. So
longer duration.
Yeah. So when we started it, we were really listening to the patients with the fatal illness do not want to be on placebo for a long time. That's one. And could we see a signal in 6 months? And we had the example of the marketed drug Edaravone in ALS, which did see an effect in 6 months. But during the trial, we learned that, we learned the Calzati story, where even in a targeted gene therapy, they needed more than 6 months. So I think it's going to be important for all ALS studies to go longer to see an effect. And then the other thing we learned, again, is about when to intervene. So we took, in the platform trial, we're taking people up to 36 months, so that we're getting some people late in the illness. And in the last 4 years, it's really become very apparent that success is going to be better early on. So besides making it longer, we're also going to change the inclusion criteria to take earlier people.
So that's an eligibility and enrollment criteria where you're trying to get more earlier stage earlier in the diagnosis into these trials.
Yeah. So earlier patients, longer follow-up. But that is the point of a platform trial, is to adapt along the way based on what you've learned.
And in terms of, like you mentioned, it's been difficult, and a lot of these drugs don't work. What impact, in terms of like from the drugs coming into the study and investment into ALS, where are we today in terms of investment in ALS, interest from industry in ALS?
Yeah, it goes up and down with kind of the success of things. I think when the Amylyx drug, which was one of the drugs that got approved on a single study, came out, I think there was a huge increase in interest in ALS by venture capitalists and companies, and again, people wanting to go for that single study approval. I think when the repeat study was negative, that put a damper on that, and so we're kind of in that valley right now of at least investment interest. So most of the companies are kind of small companies, with a few exceptions. And they're struggling for the resources to go to the next step.
So you mentioned Amylyx. Obviously it was one of those stories that, it's a fascinating story, and I covered the company for a long time. And, the drug, looked promising. It got to the market. We talked about, we'll talk a little bit about FDA in a second, but, given sort of a hallmark of FDA regulatory flexibility, and it reached the market, had essentially what was sort of like a confirmatory phase 3 study that failed, and then the drug was pulled off the market. What did you learn from the Amylyx experience? What do you think the field of ALS learned from that?
Yeah, I don't know if I'm still the only one who still thinks this, but I still think it was the right decision. And so when we designed
the right decision to approve it.
Yeah. Yeah, that's a controversial, not everyone thinks that, but again, for a fatal illness, if you have a positive phase 2 study and you can approve it conditional on doing another one, that's done all the time in fatal cancers. And this is an illness just as bad as that. And, this time it didn't repeat, but other times, it could repeat. So I think if we have another, drug that has maybe slightly more robust phase 2 studies, that's still the right thing for a fatal disease.
But I take it what you're saying, and what you think is that one of the lessons out of the Amylyx experience would not be to be, from a regulatory standpoint, to be more conservative. You think that the FDA made the right decision, that was the right call, but you would not want to see that one of the lasting ramifications of that being that they are no longer as flexible in ALS as they have been.
Correct. I think that's really important. I don't want to see them approving drugs that aren't positive in phase 2. We don't want to go too extreme, but if you have a drug that's a well-done, phase 2 study and it's positive, to have that same pathway they have for cancer. So the neurology branch actually does not have a conditional pathway approval process. They did this kind of on the word of the company that they would do another study in a reasonable time, which the company did. But I think it would be helpful to have that kind of regulatory pathway.
So we can talk about the FDA for a second. So
sure.
A lot of stuff going on at the FDA these days. Not a lot of it very good. And I wonder, you're watching this. I don't know what you're hearing in terms of sort of neurology and the neurology division at the FDA, at ALS. What are you hearing about what's going on at the FDA? Are you worried about the changes, the cuts that are, the officials that are being ousted from the agency? Are you worried about that?
Yeah, I'm very worried about it. I mean, the neurology branch, the people we work with on ALS are still there, but so many other people aren't there, so they're overworked. They were already kind of overworked before, and so I worry about the speed of response and the depth that they can review things. And then I know a lot of people are upset about Peter Mark's departure. He was responsible for at least the gene and cell-based trials that went through, and really was a strong partner and advocate for rare diseases.
Yeah, I wonder if you have any insight into sort of like what the. I mean, well, I ask you this: what are you looking for? What are you watching for at the FDA in the weeks, the months ahead? In terms of lasting impact of what's happening. Like what should we all be looking for if we want to sort of see if there's any sort of really significant change in the way the FDA thinks about these things?
I think we'd want to see if they're sticking to their, the timelines for review, if they're setting up adcoms, and they're still doing their day-to-day business.
I haven't had an adcom in a really long
I know.
time. Are you still on? Because you're a committee member.
Yes.
Which committee are you on?
I don't know what the name of the one I'm on. It's for small molecules.
But they haven't.
Yeah,
they haven't had an adcom in a while.
No, and I just rotated off, but. I'd like to see if they come out with any new policies or what their approach. I mean, I could see them going both ways. It could be more of a free-for-all that they'll approve everything, or could get so that nothing moves forward.
Yeah, it's interesting you say that because I think that's one of the things that we're all watching for is which direction do they go in? Are they going to go in the direction of where they're going to become more lenient, approve drugs on less evidence, or are they going to become more conservative? And I don't think anyone really has an idea of which way. I mean, partly is just that all the news lately has been people leaving. We haven't really seen who's going to be coming in, if anybody. So it's difficult to really kind of to determine what the future of the FDA looks like.
No, I agree. I know it because we've been working with them on the platform trial since 2020, and there has been a lot of rotation of especially the biostatisticians. And so for us, that means that things that we got agreement with, we're starting all over again every time we go there. And it's very complicated, statistics. So I do worry about their losing, the people who actually have a lot of knowledge and talent.
Mm-hmm. And then in terms of funding for ALS research, obviously, we've seen a lot of funding cut through the NIH. How is ALS? Have you unscathed? Has there been cuts? What can you? I don't know what's going on in that part of it.
So far, it looks unscathed, but obviously anything can change. And I think that's because of this group I Am ALS that was started by a patient, Brian Wallach, who used to be in the Obama administration. And that they have a very strong advocacy group that got a bipartisan bill passed for ALS funding called Act for ALS. And I think their work is, and because they have people on both sides who know about ALS and care, that somehow it's being saved. So for example, the Department of Defense announced their cuts which were drastic. They cut almost in half the research dollars, and the amount for ALS stayed exactly the same. So it goes back to that. I think that advocacy is very helpful.
Any questions? Right here in the front?
So the lifetime risk of multiple...
Actually, we're going to get a...
Benjamin Brooks, Charlotte, North Carolina. The lifetime risk of multiple sclerosis is 1 in 500. The lifetime risk of ALS is 1 in 400. And what we're looking for is actually to have more interest in terms of companies to develop drugs. What happened with MS, obviously, there was a hit, and then there was another hit, and another hit, and another hit. And that's what we need, I think, for ALS, is to maintain the interest. The other thing, though, of course, is that ALS is a military disease. It's much more common in the military, and that might be one of the reasons that DoD funding stayed, because it is an occupational risk factor for being in the military.
Absolutely, Ben, thank you. I agree on that.
Well, I guess you want to build on the success of Qalsody. So if you think about, I don't know, 1 year, 3 years, 5 years out? Where do you think the field is? Where is the field going?
I think we're going to see more of these targeted treatments for the genetic forms, but also as we get better understanding of the 80-90% of people with sporadic disease, subsetting people by biology. And I think we'll get more drugs, a bit like MS and cancer.
It would be nice at a future AAN if you have a plenary session with a real breakthrough ALS drug, wouldn't it?
Yes, we should plan on it.
Catherine, another question?
Yeah. Vishwa Vijay Singh from Azurity Pharmaceuticals. You mentioned about targeting specific gene mutations for ALS treatments, but one of the pipeline products that you showed was from Clean, the gold nanocrystals, which has a completely different way of action. So what's your take on a product like Clean's, and what's the place, how do you say that playing a role in ALS treatment?
Sure, thank you for that question. It's about a company, Clean Nanomedicine, and they have a drug, it's gold nanocrystals, that get into the brain, into the motor neurons, and catalyze a lot of reactions. But one of them is making NAD, or helping with the cellular energy. And they had very good preclinical data that, at least in cell lines, by doing that, they could improve the biology, improve the TDP-43 biology. So they've done 2 studies, and our study didn't hit on the primary, but did show a survival effect, especially the longer that we followed people. So I think they should do another study. So, and that's what they're planning, a phase 3 trial, where they treat longer and they look at mortality.
Well, we're out of time. Thank you so much. You are super generous with your time. I can tell you, as a reporter, if I have an ALS question, I call or email Merritt, and she always responds so fast, and I really appreciate that, and I appreciate you being here tonight.
Thank you for having me. Thank you, everybody.
Thank you, Adam and Merritt, for that great discussion. Up next, I will be hosting a conversation with today's presenting sponsor. Please welcome to the stage Pavneet Kukreja, Therapeutic Area Lead for Parkinson's, Medical Affairs, and Health Impact at AbbVie. Pavneet, we are so thrilled to have you joining us here today to share your knowledge and your experience. You have been at AbbVie for 15 years, 14 of which have been with the Parkinson's organization. Can you tell us what has been the highlight of that period of time?
No, thank you. First of all, this is a very timely topic because April is Parkinson's Awareness Month. And thinking about my journey at AbbVie, I have to say it has been truly incredible. One of the highlights for me has been the fact that I've been able to work directly with patients, advocacy groups, and clinicians to really understand that unmet need in Parkinson's. Parkinson's can be very unpredictable for patients' lives. It can impact their families. It can impact those everyday things that we think for granted, like going to the grocery store or mealtime. And so early in my career at AbbVie, we were creating our first Parkinson's treatment, and it was really inspiring to be at the forefront of that innovation and truly see the progress that we've made in terms of being able to provide patients with additional therapeutic options that could treat their symptoms. Before those options, I think patients really felt like they were bystanders in their life. They felt like they were living by that medication clock. They were experiencing those unpredictable offs. And even though today the field as a whole has done a lot in terms of delivering these options, we still have a lot of work to do in Parkinson's.
As you mentioned, Parkinson's is a very complicated disease, and the patient population is increasing. So how is AbbVie approaching innovation and treatment and development to meet those patient needs that you were discussing?
So yes, it is an extremely complex disease, not just from a molecular standpoint, but also from the diversity of symptoms that patients present with, and that can make developing effective treatment options kind of challenging, essentially. At AbbVie, we employ a multidisciplinary approach, and we leverage the knowledge that we've had in neuroscience for the last 3 decades. So we're really committed to understanding the molecular and genetic drivers of the onset and progression of Parkinson's disease so that we can develop novel drug targets to really tailor our therapeutic approaches. And by integrating patient-reported outcomes and advancing drug delivery as well as formulation, we really hope that we can address the real-world challenges that patients face today. And through all of this work, ultimately, our goal is to improve the outcomes for people with Parkinson's.
When you're looking ahead, what are some of the promising areas of research in the field? What are you at AbbVie focusing on when you're thinking about making progress for patients with Parkinson's disease?
Yeah. This is a truly exciting time in Parkinson's when it comes to research. At AbbVie, we're committed to expand our research pipeline for both motor and non-motor symptoms. So motor symptoms are things like slowness, stiffness, difficulty walking, and therapies today provide that relief for patients. Non-motor symptoms can be things like cognitive changes, mood disorders, sleep disturbances, and that's just to name a few. And this is largely an unaddressed area, but has a significant impact to patients' lives. When we step back from symptomatic therapy, there's a lot of advancements that have been made in neuroprotective and genetic treatments, as well as imaging techniques to really understand those biomarkers and to, essentially predict Parkinson's disease. And at AbbVie, we're actually developing disease-modifying therapies so that we can hopefully slow that progression of Parkinson's, which is a current treatment gap today because nothing like that exists. Yeah, I'm just so proud of what we're doing because we're committed to affecting patients across their disease stage and through all their disease journey. And what I really want to say is that we cannot do this alone. Collaboration is key. And this is why we engage with external experts, patients around the community, to really understand that whatever we're doing is going to enhance their quality of life and make sure we find treatments that align with their interests.
So we all know that the path of research can be really long and really challenging, and I'm curious about what keeps you feeling motivated and committed to continue your work that you were doing in Parkinson's disease research over 14 years.
Absolutely. It's the patients. Hearing their stories, seeing the difference that we make really underscores the impact of the work that we do today. Those personal stories, they get me through my hard days, but they also motivate me to continue to do everything that I do today.
You talked about the promising areas in field and the fact that patients motivate you. Is there anything in particular these days that's bringing you hope?
Yeah, listen, recently we just launched new therapy, and every day we're hearing from clinicians who have used this therapy, the positive stories that patients are able to now engage with their families, do things that they weren't able to do before, and those types of stories really, really are truly why we do what we do today.
Thank you so much. Is there anything else that you would like to add?
No, I just want to thank everybody that's in this room that's working on Parkinson's with me. I want to thank you and the STAT team for allowing us to talk about our research today. So we really appreciate this opportunity.
Awesome. Thank you so much.
Thank you.
So you all are probably aware that for the first time in decades, there is a new drug for schizophrenia on the market. Here to tell us more about that, please welcome to the stage Dr. Sumit Hirawat, Executive Vice President, Chief Medical Officer, and Head of Development at Bristol Myers Squibb, and to moderate, Stat Biotech Correspondent Meghana Keshavan.
Hey there, it's lovely to chat with you on a stage.
Yeah, thank you, and thank you for providing this opportunity and to talk about it.
Yeah, no, and before we begin speaking more broadly, I was wondering if you could just tell me what is Bristol Myers Squibb's approach right now to the broader psychiatric space?
Yeah, and maybe I can take a step back, and before we launch into neuroscience and neuropsychiatry, just the approach to diseases. And as you know, we are in the heme space and oncology space and cell therapy space and immunology and cardiovascular disease, and of course resurgence into neuroscience. And as we think about overall, there's a lot that is happening as we look even in 25 and 26, as we look towards the back end of the decade as well, with obviously Cobenfy being a really, really important molecule and the medicine that is now approved for patients with schizophrenia. Now that molecule was approved last year. We have more readouts this year as we think about just within a couple of months for adjunctive schizophrenia treatment. And then of course at the back end of this year would be the ADP or AD psychosis. And then every year thereafter, we may have a new indication of reading out because we have 7 new phase 3 studies starting just this year. And then, of course, in 2026, from a pipeline perspective, there's a lot that is happening, potentially 6 new molecular entities that will read out across the therapeutic areas. But coming back to neuroscience, and I'm sure we'll go deeper into it, our approach is not just neurodegeneration or just a neuropsychiatry, but a holistic approach to think about modalities, about patients, about improvement in the disease, about improvement in the symptoms, and how to also capture data in a way that we can develop new endpoints of the future through AI and machine learning.
Very cool. Yeah, so just going back to Cobenfy, it was approved last year after, again, last year you acquired it for $14 billion, the company that made it.
I don't know the numbers, but sure.
I think so. It was a lot. But Cobenfy has been really interesting because it's a new treatment modality for schizophrenia, and there hasn't been a new approach to this disease for quite a long time. Why did you decide to pursue this as a target, and what potential does this new mechanism of action have? Can you, first off, can you tell me why it's different and what else it could do?
Sure, and certainly the audience knows it very well that in schizophrenia in general, there are lots of atypical antipsychotics that are approved, and primarily dopaminergic pathways are impacted. The reason the acquisition that we had of the company called Karuna, and specifically the KarXT, is because it has a dual muscarinic agonistic effect. And of course the scientists were very smart to also combine that with trospium, which is a peripheral system blocker of that muscarinic effect. And therefore you can manage the side effect profile better. So over the last 3 decades, as you just said, there was no new mechanism of action that came by. Now, we had been watching it from afar, the Karuna story evolving and developing. As you know, neuropsych, and I think earlier it was said as well, it's a difficult space to develop drugs. Why? Because there's no specificity in terms of the causal human biology in the neuropsych space. Now, in neurodegeneration, yes, you have mutations that you can go look for. There are certainly genetic markers that you can look for, but in neuropsychiatry, the neurotransmitter systems, they are harder to recapitulate. And therefore, the de-risking part that occurs during clinical trials occurs pretty late. So we were watching Karuna, and as soon as, of course, the emergent trials started to read out, we, of course, got in a room and talked to them, and it was a very, very good discussion, sitting across the table, trying to solve the problem, trying to understand what is it that we can do. And once we had the drug in our hand, of course, we've now launched many, many more indications that we'll be looking at going forward.
Yeah, no, it's really interesting. The trospium xanomeline combination is cool because I know that it was tested, the xanomeline itself was tested decades ago for Alzheimer's, and then got shelved because of the side effects you mentioned. That seems pretty common with a lot of psych drugs in general. Like, there's potential, and then they just don't work, and you just get this, like, we were talking, what was it, informed serendipity in terms of like identifying actual psychiatric drugs. So I guess I was wondering, like, since it's such a risky space, and pharma has backed out of it for decades, and now is suddenly interested again, why did you decide that you wanted to get into neuropsych again?
So look, our history with neuropsych is not new. It's 20 years ago, Abilify was one of the largest products that BMS had. And we always had the intent, so after the acquisition of Celgene, we were working in this space in a stealth way. We were working through external collaborators, trying to find new molecules, but primarily in the neurodegenerative space. So in the Alzheimer's disease space, working with a company called Prothena, we were working on the anti-tau molecule with them. But there were other places where we were looking as well. So Karuna was certainly on the radar, and we certainly knew the unmet medical need is very high, and there is an opportunity to bring something new, and Karuna provided that opportunity. But we're not stopping there, because we know what we just heard earlier, mental health challenges are there. So what can we do to certainly continue to improve on that? So beyond the CAR-XT example that I talk about, and we're launching the bipolar manias phase 3 studies now. We're launching the Alzheimer's disease-associated agitation studies this year. We're launching Alzheimer's disease-associated cognitive impairment studies this year. Next year, we'll start the autism program as well. But behind that, we have other programs for disease-modifying agents. So anti-tau, the MTBR region-targeting anti-tau molecule, that's already in randomized phase 2 studies, and in 27 that will read out. We have a molecule that is going into the phase 2 randomized studies targeting dual inhibition, Fa, which is the two enzymes, Fa hydrolase and the MAG lipase. These are two enzymes that are critical in terms of the endocannabinoid metabolism or degradation. So those inhibitors, that is going into phase 2 studies in spasticity associated with MS, as well as in agitation for Alzheimer's disease. So we don't want to stop. And our certainly commitment is there in the neuroscience space, and we want to continue to further that.
Sure. And in terms of this, what was it, the FAAH?
And MAG lipase, yeah.
Yes, that one. Targeting the endocannabinoid system. That was inspired by marijuana, kind of, wasn't it? Like the impact that has.
In a very crude and way, you can say that. But the biology is quite fascinating. Look, I'm an oncologist by training, and so we do all kinds of crazy stuff, and we've really gone far and looking at all modalities. But neuroscience certainly has been a fascinating field, and I continue to learn, and as I read more about it, many companies have approached it as either a singular inhibitor of the hydrolase, the fatty acid hydrolase, or monoamine glycerol lipase. But I was there at a conference last year, and we were talking to a scientist who was presenting, and when I started talking to her, she goes, yeah, in the mouse models, what we've seen, if you can inhibit both these enzymes, you can really calm the system down. And we were already doing the preclinical work, and then we had already gone into the humans for single ascending dose and multiple ascending dose studies. So when I came back, we started talking about indications, and now we are launching the randomized phase 2 studies. So you learn, as you learn, as you talk, and ALS was a great discussion that we had earlier on. So we have a program that we are in the infancy stage of thinking about that as well. So we'll continue to grow through there, and that is why collaborations, I think earlier somebody else had mentioned, collaborations are going to be important, and we have to develop those.
Sure. And why is the Cobenfy molecule so special? I'm just wondering for schizophrenia, for Alzheimer's, for other indications like bipolar. I'm asking it because even though it targets the muscarinic receptors, I think it ultimately helps modulate dopamine in the brain, which is similar to the other antipsychotics on the market. So why is it distinct?
So I think the way I think about it and the way science tells us, first of all, dual muscarinic agonism is important. So M4 and M1 muscarinic receptors are the ones that are targeted, and it's a direct agonist. It does not require a milieu or, for example, acetylcholine in the system. So since it's a direct agonist, you start to see the effects also in the periphery, and that's why the trospium was very important. So one, you have a profile that from an efficacy perspective gives you what you want from a positive symptoms, negative symptoms, and in a subset of the population that we have published, we also see cognitive impairment changes. So you start to improve those, and we publish the data. Based on the xanomeline study that you talked about, that is the published work, and there are 6 other studies that were never published, but we now can see the data because we now have the databases. There is also impacts on improvements in other symptomatologies, including the mania part of it. So that's why launching the bipolar mania makes sense. Now, of course, one of the key elements is, okay, today, patients with schizophrenia are treated with dual atypical antipsychotics. There is no approval. There's no reason to really combine them, but you really do want to help the patient to see if they can actually improve, and you can improve the efficacy. But we do know that there is a side effect profile that they carry as well: weight gain, hyperprolactinemia, movement disorders, akathisia, etc., which we did not see with of course, in our molecule, that is Cobenfy. So, but we are doing that study, and hopefully the study reads out with the data that is going to be convincing that we can put Cobenfy on top of atypicals as well, so that patients can benefit more. And then, of course, the rest of the story that I already told you about the other indications as well.
Yeah, and just speaking of other indications in neuroscience or neuropsychiatry, and as you're expanding, Alzheimer's has obviously been huge in recent years with recent approvals like Leqembi and whatnot. You have this tau targeting molecule that you're working on. How does that differentiate itself, you think, from what's already kind of beginning to exist?
Oh, absolutely. With a lot of care. I think earlier it was said, in neuroscience, we have to go back sometimes to go forward, 2 steps forward, 1 step backward. And certainly, a lot has been learned through the non-successful development of many tau-directed therapies. And the whole protein has been targeted in various ways. So where we are approaching it, and I guess there are 2 molecules, one that is ours, one is a competitor molecule, looking at now the specificity of the MTBR region, the microtubule binding region. So there are 4 areas, R1, R2, R3, R4. We are targeting R1, R2, and R3. The competitor molecule is R2, R3, R4. So we'll see if that helps us in terms of degradation or removal of the tau pathology that we have. But of course, there's a lot of work that needs to be done, and it is a very challenging area because identifying the patients, not clinically, but also who are biomarker positive, it is a humongous challenge and a huge and a very, very steep mountain to climb, because you go do these trials, but as you start screening the patients who are clinically diagnosed as Alzheimer's patients, as soon as you do the biomarker work, 50% of them don't have a positivity for biomarkers. So there's a lot of problems to be solved, and we are working on that right now.
Okay. And just hopping over to multiple sclerosis, because I know that you're in that space as well. What's your approach in that indication? So
we've taken a holistic approach wherever we think about a patient, right? We're not just talking about treatment of a disease, but also the symptomatology, and that's why I talked about earlier. So we've got Zeposia that's approved for multiple sclerosis. That's a treatment. You've got pharmaglipase going into the spasticity associated with multiple sclerosis, so we'll talk about that, or we talked about that. But then we have also the area of approach from a cell therapy perspective. And there's a trial that is ongoing. I think some data were presented from competitor on 2 or 3 patients from 2 different trials looking at MS through cell therapy approach, CD19-directed cell therapy approach, so that you can remove the B cells and you can impact the disease. Of course, it's very early right now. We presented safety data for 4 patients earlier this year. We'll have more data coming out in a month or 2 on slightly more number of patients. But as was said earlier, neuroscience is a disease where neuroscience is a science where diseases take a long time to be followed and to show if there is a benefit that we'll achieve, especially in the neurodegenerative space. Mm-hmm. And so we have to follow these patients for much longer, although it's a one and done therapy, but we'll have to follow longer to see if we can really impact the disease in terms of looking at the bands as well as the neurofilaments, if we can get removal of those.
Sure. And then I was just kind of wondering, what do you think is cool out there that that you have your eye on, that you don't need to disclose anything that is private, but Like just in the broader neuropsych space, I'm just wondering, are there any developments that you're just intrigued by, and as a person?
As a person, I would say, and you know, neuropsych has a lot of space to improve, because again, what we are seeing really is a molecule that has a different formulation that comes out, or a molecule that has a tiny bit of a change from one isoform to the other, and that comes out, but they're all targeting the same way. And I think we have to change that philosophy. Now it's, again, not easy, but we'll have to continue to move forward. Again, at one of the meetings, I met somebody who was talking about the whole brain stimulation through insertion of these prefrontal cortex rods. But then there are companies that are working on different ways of doing that with the devices that are not as invasive. So that's pretty cool. But there is a lot of work in the depression space, anxiety, generalized anxiety, which is continuing to grow, in fact, in the world with everything that is going on. What can we do about that? And I think there is a lot of work to be done over there. But my passion certainly is in the neuropsych space and ALS, as was said earlier. So we are continuing to work on that area and see where we can go with that.
Sure. And Adam beat me to this question, but I got to ask it. You know, things have been changing in the government, in the FDA, regulatory processes. How is that affecting Bristol-Myers Squibb?
Yeah, it is concerning to see what is happening. Nothing that is changing is for the good from a regulatory perspective. Why? Because you want people who are knowledgeable in the regulatory sciences. People who are not only knowledgeable but responsible, understand the disease, understand the process. So as was said earlier, if you don't have enough people, what will it do to timelines of just investigational new drug applications, for example, to get a safe to proceed letter? What is going to happen to the new drug applications or the biologics applications that we submit? What is going to happen to inspections of facilities? If you remember, during the COVID times, inspections could not happen, therefore drug approvals got delayed. Do we want that again? Do we have people who understand policy enough to really negotiate the next PDUFA 8 that is coming through? So these are very, very critical elements that have to be understood, and therefore you need the right people. There's a lot of good people that have already left, and so we have to continue to pay attention to that. From a BMS perspective, certainly the primary focus is the science, but science cannot progress further without regulation, and certainly free-for-all will not be a good idea either. So we remain concerned, but certainly we do not stop. We continue to move forward.
Sure, and I mean, I was actually wondering in that vein, I mean, what is your timeline in this space for the next 18 to 24 months? Like, do you have, I mean, you have trials underway in neuropsych. Are they affected by the changes right now? And are you still pushing on as, you know, what do you have coming?
Well, there's a lot, as I said earlier. So our whole motto is to accelerate to achieve 10 new molecular entity approvals and 30 other indications approved by the end of the decade. And we are starting with a bang, because this year itself, we have several phase 3 readouts, both for Cobenfy as well as for another molecule, Camzyos, another molecule, Reblozyl. Then next year, we have 6 new molecular entities reading out phase 3 trials. In multiple myeloma, we've got 3 things reading out, 1 cell therapy, 2 cell mods. We've got Melvixian that is reading out next year as well. We've got Raise Bio molecule, which is the radioligand therapy, reading out next year. And there will be obviously more readouts for Cobenfy as well. So we have a lot to deliver, and therefore getting to the right place from a regulatory perspective, not only just in the US, but how it impacts the rest of the world. We have to keep that in mind, because there is disease that is around the world, and we have to serve everybody. We conduct clinical trials in 45 countries around the world.
Yeah.
So we really have to live up to that promise that we make.
Yeah, for sure. And I apologize, audience. Does anybody have any questions?
I think it's crystal clear.
Everyone understands. Yeah, but I mean, I was also just wondering, what is your intention here at AAN? Like, are you looking for partnerships?
Look, I think what we just talked about over the last 20 or so minutes, it's pretty clear what our mission is, what our passion is, what our purpose is, and we really do want to impact, through our commitment in neuroscience, the outcomes for patients. We've got a pipeline that is broad, there's a pipeline that is deep, and certainly, as was said earlier, this cannot be achieved without collaborations. So we're open for business. Physicians, patients, advocates, you want to come talk to us, please come talk to us.
Well, thank you for talking to me.
Thank you.
It was really lovely, and I'm a huge neuroscience nerd, so thank you.
Thank you very much.
Thank you both for that great discussion. Up next for our final conversation on GLP-1s in the brain. Oh, sorry, we have to do the stage change. But we will be welcoming to the stage Scott C. Bunce, who is the Associate Professor of Psychiatry and Behavioral Health at Penn State Health and Penn State College of Medicine, Paul Edison, Professor of Neuroscience, Division of Brain Sciences in the Department of Medicine at Imperial College London, UK, and Head of the Imperial College Memory Research Center, and to moderate, Stat National Biotech reporter Elaine Chen.
So everyone's probably heard of Ozempic, Wegovy, the GLP-1 drugs that have become wildly popular. They were developed initially for diabetes and then obesity. And the thought was originally that they work in the gut. I mean, the GLP-1 hormone was initially discovered as a hormone that promotes insulin secretion in the pancreas. But it turns out a lot of their effects on weight loss is actually probably occurring in the brain. And these drugs are probably doing actions in the brain that we're still understanding to help curb food cravings. And it turns out they might also help with a lot of other neurological conditions. So that's what we're going to talk about today. I would like to ask both of you to start with, how did you get interested in studying GLP-1s for Alzheimer's and addiction? How did you even think about that and learn about it? And give us an elevator pitch on why you think more people should be studying this. So Paul, you want to go first?
So, yeah, thanks for inviting me. So if you were to actually look at what happens in Alzheimer's disease, one of the key things which was striking to me was apart from amyloid, you got several other pathological processes happening. So you got tau formation, neuroinflammation, insulin resistance, astrocyte activation, and so on. So we did one of the first amyloid imaging along with the Pittsburgh. However, I was also working on inflammation, tau, and astrocytes, and we could see there's a clear disconnect between these pathological processes. And we know that the glucose metabolism goes down the medial temporal lobe, and we know that the amyloid deposition starts in the frontal cortex. So, especially after the first anti-amyloid agent, which was produced by Ilan Wyeth way back in 2004, it was very clear. We cleared the amyloid, but the FDG went down significantly. Even at that time, so even though amyloid theory was very strong, we thought, hang on, amyloid alone is not enough. So that's why actually we started looking at the compounds which can influence other pathological processes. So my view, even at that time, was very clear that you need to have multiple drugs or have a single drug with multiple effects. And that's why actually I bumped into the GLP-1 analogs. So if you were to look at how it works, it has got multiple effects, and these multiple effects are probably the reason why it might work in Alzheimer's disease.
But tell me how you got interested in it, because you just told me earlier that you actually reached out to Novo Nordisk, which is the company that makes Ozempic, 15 years ago, about studying it in Alzheimer's. So that was probably even before they really started seriously developing it for obesity. So tell me about how you got interested in it that long ago. And I mean, Novo Nordisk now is running 2 phase 3 trials testing Wegovy in Alzheimer's. So they eventually got on board, but it was a long process.
Yeah, so I think the key. I was particularly interested because of the multiple actions. From the industry point of view, they were very much focused on diabetes, and they were not really interested in looking at Alzheimer's disease. So it took a bit of a persuasion, to say the least, to get the drug and placebo, and that's how we actually started the clinical studies in patients with Alzheimer's disease. Clearly, some of the findings which we had, so we actually looked at about 204 patients. They all received liraglutide 1.8 milligram for a year, and we had the FDG as well as the MRI scans and detailed neuropsychometric testing. Even though the primary outcome of the study, which I briefly presented yesterday, was negative, what we have clearly shown is the cognitive outcome measure, as well as the MRI volume was preserved in patients treated with the liraglutide. So there's clearly a signal. While we were doing this study, there were so small studies were coming out showing, okay, there may be some signal in the FDG, there's some signal in the fMRI studies, and then there came a huge number of observational studies based on the databases. So clearly there is some signal now.
Okay, I'll come back to those studies because there's a lot to dig into. But Scott, I want to come to you. You've been looking at GLP-1 drugs in addiction. So tell me how you got interested in that. Where did you even start thinking about running some trials in that area? And give us your pitch on why more people should be looking at this.
So I was actually brought into this area by my multi-PI in the project, Dr. Sue Grigson. And for those of you that don't have, I think everybody knows that we're been in an opioid crisis for 12 years. And to give you a sense of what that looks like, last year we had 8.9 billion individuals in the US over the age of 12 misusing opioids at something like $350 billion to the economy. So Sue had been looking at this. The other important part of this is that right now we have 3 FDA medications approved for treating people who have an opioid use disorder. In perspective, we have 7 classes of drugs that we use to treat depression, 30-some individual drugs. And so one of the important things is that the drugs that we do have all work on the mu opioid system. And Sue thought that there might be a better approach because there were a lot of problems that these medications were not taken care of in folks, and so people were dropping out prematurely. We didn't have the kind of coverage that we needed. And so she started looking at this in terms of a mechanism of, instead of addressing the pleasure system in opioids, and in most addictions, it's looked at as that, we're looking for gratification, is looking at the satiety system, which the GLP-1s were showing. And so she did some initial studies in animals, in rats, and showed that that liraglutide, which is also known as Saxenda, if a rat was on heroin and you gave them liraglutide versus a placebo, it knocked down their use. And that was true whether they were given a cue, that might be, say, like a picture of a beer or a beer commercial, or whether they gave them drug, like giving somebody a few drinks of alcohol, or if they stressed them, so they gave them euhimbine, which is, not unlike talking to a friend that you don't like.
And sorry to cut you off there.
Go ahead.
But I want to get into, because you actually did run a trial in humans.
Yes.
People with opioid use disorder. So tell us a little bit about what you found there. That was a small trial, but you also
Yes.
are running a bigger trial now. So tell us about that.
So yes, so while Sue was finishing up some of those animal studies, we also ran a clinical trial. We did that at the Care and Treatment Center in Wernersville, Pennsylvania, which is a very high-end facility. And we were able to put people into this, it was a full, double-blind, placebo-controlled clinical trial. And we were able to put people on either liraglutide or on placebo. And they went through the rest of the treatment as anybody else would. One of the things that was, they were allowed to pick whether they were on buprenorphine or not. So of the 20 individuals that we got in the study, 13 of them were on buprenorphine, seven of them were not. We had 10 and 10 in each study, or each in each group. And what we were able to show, importantly, was that across all people for all days that they were in the study, that its medication dropped their craving by 40%. What's another important part of this is that all these medications, GLP-1s, because of their effect on the GI system, they're ramped up over time. So you start with a lower dose and you keep going up. That's a bit of a problem if you're doing treatment for substance abuse, because in particularly opioids, because you want to get people on a treatment very quickly and you want it to work so that they're protected. And what we also found was that this was effective at the lowest dose.
And
at
also,
the starting dose.
The effect started very quickly, right?
Yes, and yes, in subsequent analysis, we actually found that we've had effects by day 2, which is really important, because that gives us a sense that the person's going to have some safety. The way we did this is we had people, we gave them smartphones, and they were programmed smartphones, and we were able to measure 3 or 4 times a day what their craving was, what their mood was, what their, and we were able to see that by day 2.
Yeah.
So that
Which
was
is that's very quick, considering that for weight loss purposes, I mean, the weight loss doesn't start until much later on. So coming back to Paul with the work that you're doing in Alzheimer's, like you said, the study that you ran was kind of mixed results because it didn't meet the primary. The study that you ran in about 200 patients, testing an older GLP-1 drug in Alzheimer's patients, it didn't meet the primary endpoint of brain glucose metabolism, but you did do MRI scans and you did see that people on the treatment had less shrinking in parts of the brain involved in things like memory and learning. I guess it's not yet a home run, right, for GLP-1s and Alzheimer's, and also similarly for Parkinson's. We're still in we still don't really know yet. The results so far are kind of mixed.
Yeah, that's absolutely correct. So I think, what we have shown is there is a beneficial effect on the MRI as well as the cognition. For whatever reason, this is the first study where there is a divergence between the MRI volume and the FDG PET. So what you normally see is the FDG and the MRI correlates very well. We're trying to look at what exactly is the reason for that. What you have to bear in mind, even though we had about 202 patients randomized, the total number of subjects are relatively small for an Alzheimer's study. And most of the studies, if you were to look at, you need about thousands of, at least 1,000 patients to see beneficial effect on the cognition. So while it is not the perfect study in the sense, perfect results, I would say seeing some improvement in the cognition as well as the MRI volume is quite encouraging. You have to consider this as a mechanistic study. So what we are trying to say is there is some level of target engagement happening, even though the mechanism by which the GLP-1 analogs works is still being debated. The same with the Parkinson's disease. There are some studies that have shown some beneficial effect, some haven't shown beneficial effect. And then there is also the key issue of how much of these drugs are getting into the brain. Some of these drugs don't get into the brain. Some of them do get into the brain, as I was highlighting yesterday. So if they're having a significant systemic effect, that will, along with the central effect. So my belief is these drugs work both from its peripheral effect as well as the central effect. And how the peripheral effect influences central effect, which we need to understand better. And we also need to understand how exactly the central effect is happening.
Oh, so that's interesting. So you're saying potentially the way that it could help with Alzheimer's is not just its direct effects in the brain, but also the way it works in the body that influences that like signal, that's the connection to the brain.
Absolutely. I mean, so if you have got a beneficial effect on the, say for instance, reducing the insulin resistance peripherally, also having the influence on the diabetes, obesity, influence on the heart, kidneys, blood vessels, and the whole cardiovascular outcome trials in GLP-1 analogs clearly demonstrate significant benefit of the cardiovascular risk. So that will have definite impact on the central nervous system. Now, what is the mechanism by which it is happening? We still need to clearly understand. But along with that, we know that the GLP-1, once the GLP-1 receptors are activated, it activates the cyclic AMP and AKT pathway, and that itself causes the, improved neurotransmission, reduced inflammation, and it also has got a separate effect on the microglia as well as the astrocyte.
Just a reminder, if anyone in the audience has questions, you can just raise your hand. So you mentioned an interesting point, which is maybe a limitation with the current treatments we have is that they are peptides, so fairly large molecules. We do have small molecule GLP-1 drugs that are in the pipeline. They're not yet approved. Do you anticipate we kind of would see even greater effects on Alzheimer's, on addiction with the small molecule GLP-1 drugs, assuming that they would have greater brain penetrance?
Yeah, so I mean, at least from the pharmacology point of view, if they're entering into the brain, and from what we are seeing so far, it should have better effect. One thing you have to bear in mind is if you are looking to treat large number of patients, it has to be oral tablets. Yeah. So I think that is one of the key things, and that's what I was trying to highlight yesterday, is that these drugs have the potential to become like the next statins, provided it can be used in a quite easy, i.e. as oral tablets, because daily injections or infusions and those things are not going to be that easy from the majority of the patients.
Scott, I want to come back to you briefly, and I want to get more into the mechanism. So what do you think are the biological mechanisms that may explain why GLP-1 drugs could be helpful with addiction? Do you think it's similar to the way they curb food cravings?
It's a very good question. I won't say that we have anything close to the real answer yet. I do think that it makes sense that in talking with my patients, their phenomenological experience is that it just shuts down all the noise. It doesn't necessarily make sure, it allows you to kind of sit back and make a good decision, a healthier decision.
And that's interesting because that's similar to what I hear from people with obesity, because they talk about food noise. And they also say, it just gets rid of that food noise.
And so what the exact mechanism is, we know that this is part of the whole system, and the way I like to explain it is you're reaching for that second piece of pie on Thanksgiving, and you go, I don't really need that. But getting that, in a sense, before you've eaten the turkey and all the other stuffing, et cetera. So just that sense of satiety is something that is really important. Now, whether that's coming from a central source, still up in the question in terms of humans.
Oh, we have a question.
Hi, my name is Julia Margulis from Blue Matter Consulting. I'm interested in this conversation we've been having, sort of comparing and contrasting the effects of GLP-1s on obesity versus some of the neurological conditions we've been discussing. And for example, this idea that the impact on addiction can be very fast versus the impact on weight loss can take a while. I know we also see that patients that come off GLP-1s tend to regain the weight, right? So I'm curious how you both think about the impact on neurological conditions. So, is the anticipated sort of treatment regimen kind of a chronic treatment, because you would anticipate that if a patient discontinued, they would regain the weight, in quotes?
Yeah, excellent question. And so what we can tell from the rat models is if you stop the medication, they start back on the drug. So that is important. Now, the thing that's different about humans is that once you get them to the point where they're not using and not craving and that's not part of life, you can help them try to build in methods and ways. This is where the behavioral counseling comes in as part of it. Just like with eating, if you stop the medication, are you going to get that hunger back? Seems like you do. What can you do to prevent that? Build in the healthy eating habits before going back off of it. So now, on the other thing, do you tell a type 1 diabetic to stop using their insulin because they've used it long enough, right? It's like, no, it is a lifetime need for insulin. And addiction can be viewed in a very similar way, that what is wrong biologically. You're not going to fix it by saying just stop.
I think we have another question.
Hi there, Paul Monis from IBG Health. I'm going to ask for a little wild speculation here. There's some novel ideas being tested in Alzheimer's and addiction, GLP, but some others including clinical psychedelics, specifically ibogaine for opioid addiction. I'm wondering if you both could comment on a little bit of kind of blue sky future casting. What is the landscape for the conditions you're treating? What are the new ideas percolating up now that are going to reshape the conditions that you're focused on in the next 5 to 10 years, like GLPs and metabolism, like maybe psychedelics or anything else? Thank you.
So if you were to look at the neurodegenerative diseases, so one thing is very clear: it is not a single process or pathological process happening. So if you were to look at Parkinson's disease, Alzheimer's disease, Lewy body dementia, frontotemporal dementia, they all have got quite a lot of common pathological processes happening. And I fully agree with you, whether we are going to lump them all together as one disease. And that's clearly, I was implying when I said, this is going to be a new statin. So potentially GLP-1 analogs can have influence in most of the neurodegenerative diseases. And when we say the patients with Parkinson's disease, the preclinical models, they just tested only the dopaminergic neurons. They didn't look at what happens to inflammation and various other things. So we don't believe that this is a specific disease-dependent mechanism. It is a general mechanism by which it is having an influence on the neurodegenerative process. Now, what happens in addiction? I haven't been working with addiction, but the reality is it has got a significant influence on the satiety, and that in one or other form influences the addiction, and that is my sort of a take on that. So I think it provides an opportunity for the neurodegenerative diseases in general. And the reality is, and that's why I was going back to the idea that if we have got an oral compound which gets into the brain and is also able to influence the whole system, then it can be like you can use that to prevent neurodegenerative process as well as treat, pretty much similar to statins. So you take the statins when you've got a cholesterol, and even if you have a stroke, you continue to take the statins. And that is going to be the future, and I don't think we are too far away from that. Clearly, there's a lot of work needs to be done because there's only one large-scale study is happening with the GLP-1, which is by the Novo Nordisk, two studies in one, so to speak. But apart from that, there's no large-scale studies, and that's what is necessary, both in the field of prevention as well as the treatment. Thank you.
Scott, are there any wild ideas in addiction you want to talk about?
Well, I think I want to quote Douglas Adams here, who said that it's difficult to fix most complex problems with just potatoes. And I say that because even now, the NIAAA has for several years been working on trying to come up with descriptions of different types of alcoholic use disordered individuals. Why? Because they ain't all the same. And as a consequence, they say, why do we have 7 classes of antidepressants and 30-some medications? Why? Because people they're coming to that in different ways. And I think that's the same thing that we need to address. It's an important question. I think we need to address the fact that there are different reasons, and working clinically, I know that there are very different reasons for why people are sitting in my office with a history of substance abuse. And some of them, it's serious trauma. Some of them, it is other feelings. Some of them is, I've had people tell me, I tried a opiate because I got an injury in my 20s. It was the first time I felt normal. So there are a lot of different things, and I think there's room for these medications, and there's room for understanding how to use those medications within one's life. They're going to make a difference, but it's a great question.
Audience question.
Hi, my name is Jiyoung Suk, Neurocrine Biosciences in San Diego. You mentioned, Dr. Edison, about the incretins or GLP-1 needing to go to the brain to see cognitive effects, but that the effect of those molecules that are in the trial, despite they didn't go very well into the brain, that they might have both brain and peripheral effects combined. And that might also be a mechanism for the weight loss. So my question is, in geriatric patients where too much weight loss could be an issue, if you had more brain-penetrating incretins or GLP-1 agonists, what are the safety concerns and what would the dosing regimen look like?
Just to add on to that question, because I did want to ask that, for opioid use disorder patients, they typically are underweight. And then, with Alzheimer's or Parkinson's patients who are older, we are concerned about loss of muscle mass, frailty. So for both of you, how do we deal with these concerns about too much weight loss for these populations that you're testing in?
Yes, so I think, let me tell you about the results of the data. So we had about 204 patients, half of them did have, active drug. So we saw about, at the maximum, 5 kilograms of weight loss in these patients. Most of them are underweight or normal weight. So we didn't see that huge amount of weight loss, which we see in obese patients, partly because the mechanism by which it acts is, once the weight as well as the blood glucose comes down, these GLP-1 analogs actually stop the influence on the weight loss.
There's a plateau.
Yeah, basically. So I think in real life, we are seeing that. And I must say, I was very concerned when we started because we excluded patients with the diabetes. So none of our patients had diabetes. So I was very concerned, we're going to give them this drug and they're going to have hypoglycemia and they just fall over. And the reality is none of them actually had hypoglycemia or significant. Some of them did have about, 6% of the patients had some weight loss. But none of them actually, I had to take them off the study because of the weight loss was excessive. The real side effect was the nausea and gastrointestinal side effects. And we had a couple of patients, 2 patients, we had to come off the drug. Within within days, they have got the nausea. And that's why probably I was saying, the influence on the addiction is partly because it is so potent. It has got that nauseating effect. So, yeah, I'm pleased to see that they didn't have a significant amount of weight loss. Yes, if we have got a potent agent, especially small molecules, which we haven't been tested before, we need to see how that behaves. But I'm quietly optimistic that that may very well work without a huge amount of side effect.
Scott, do you want to
Sure.
just very brief comment?
Very brief. And we also had these as exclusion criteria if they were below a certain weight level, because actually this is part of what we see in the medication, and when they tell us, we actually read those little slips that came with it. But I think that so you can work with individuals that and again, this is going to be careful monitoring of a side effect essentially. Now, if you're taking it, it's not a side effect if you're taking it specifically to lose weight, but it is a side effect if you're taking it for another use. And so it's something you can monitor and be aware of, because it's not, it's not an absolute have to have kind of effect, because you can eat. And people have talked about the fact that it affects the way they taste their food, et cetera. And so it's something that I think is manageable. If somebody's dropping a lot of weight and it's a problem, just like any other medication where you're seeing a deleterious side effect, you need to pull them off it, and that medication's not going to work for them unless they can. But you can feed yourself and keep the calories on if you want to.
Great. Okay, we're out of time, so we're just going to end there. Thank you both so much.
Thank you.
Thank you.
All right, thank you to all of our speakers and to all of you for being here this evening. Before we end, I want to highlight a few of our upcoming events. The Stat Breakthrough Summit West is right around the corner, happening in San Francisco on May 14. Some of our speakers include Reed Jobs, who is Steve Jobs' son and the founder of biotech venture capital firm Yosemite, which aims to make cancer non-lethal. We also have Aviv Regev, who will join us to tell us how she is using AI to reimagine research at Genentech, and an interview with Karen Knudsen, the former president of the American Cancer Society and newly appointed Chief Executive Officer of the Parker Institute for Cancer Immunotherapy. We're also going to be in Chicago for ASCO, and we'll host an evening event there on May 30th, open to everybody who's in town for the weekend. And lastly, our flagship Stat Summit will take place in Boston on October 15 and 16. More information about that event and all of our events is on our website, statnews.com. Thank you again to our presenting sponsor, AbbVie, and to all of you for joining us here this evening. We will have the space open and available for the next 30 minutes or so. Feel free to stay, get some food and drinks. You can head over to the bar and to the beautiful patio outside. Thank you, everyone, and enjoy the rest of your evening.